Hi everyone! Today I'm going to show you how to perform the gram stain through a step-by-step lab tutorial. Before we begin, let's review what you should be able to do after watching this video. By the end of this video and lesson, you should be able to listen and explain the function of each gram stain reagent, as well as identify the correct sequence of performing the gram stain. This is the overview of the procedure of the gram stain. First, primary stain, which is crystal violet. Second is the mordant, grams iodine. Third is the decolorizer, ethyl alcohol. And finally, fourth, the counterstain, grams safranin. As I demonstrate each step, I'll explain not just what to do, but also what is happening at the microscopic level inside the bacterial cells, so you can understand why each step matters and how the differences in the cell wall structure affect the final results. Before you begin the gram staining process, remember that you'll need to have a prepared bacterial smear on your slide. In this video, we'll focus on the gram stain itself. For this demonstration, I have two bacterial smears prepared: E. coli, which is a gram-negative bacteria, as well as Staph aureus, a gram-positive bacteria. Before we begin the staining process, here's what bacteria cells look like under the microscope when they're unstained. Whether they're gram-positive or gram-negative, at this point the cells really have no color. They just appear as clear shapes against the background. You could see their basic forms, but it's almost impossible to tell them apart. That's why the gram stain is so important. It helps us highlight the differences between these types of bacteria. The first step is to add crystal violet, the primary stain. This stain covers all the bacterial cells, turning them all purple, no matter what type they are. The stain is let to sit for 2 minutes. That way it can be absorbed by the cells. At this point, both gram-positive and gram-negative bacteria will be purple. Once the crystal violet has been on the slide for about 2 minutes, we gently rinse it off with distilled water. Included is a microscopic view of the bacteria and what it might look like at this stage after the primary stain has been applied. The next step, step 2, is the application of the mordant, which is grams iodine. Grams iodine helps to intensify the primary stain, crystal violet, and it also acts as a killing agent. We let the grams iodine sit for about 1 minute. At this point, gram-positive and gram-negative cells will both still appear purple and maybe even look a little darker purple. After letting the grams iodine sit for about a minute, it's time to rinse it off with distilled water. On the left-hand side is a microscopic view of what each bacteria would look like after this step. The next step is the decolorizer, using ethyl alcohol. This is considered the most critical step in the gram stain procedure, and I'll explain why. The purpose of decolorization is to take the primary stain off of gram-negative cells only, while gram-positive will remain purple. This is a key step in order to differentiate gram-positive and gram-negative bacteria. One thing that can happen is over-decolorization. This is when you use too much decolorizer or the decolorizer is applied for too long. When this happens, you risk gram-positive cells losing the primary stain and appearing red and pink, giving false gram-negative results. Under-decolorizing occurs when too little decolorizer is used or the decolorizer is applied too briefly. When this happens, gram-negative cells will retain the purple stain, giving false gram-positive results. This is why it's important to apply the ethyl alcohol or the decolorizing agent drop by drop until the stain runs clear from the slide. Notice how the microscopic view depicts E. coli as colorless. Rinsing with distilled water immediately after will stop the decolorization process. In this case, when looking at the microscopic view, we saw that E. coli became transparent, while Staph aureus will remain purple. The final step is application of the counterstain, gram safranin. This is the final reagent and is used to stain the decolorized cells pink. The gram safranin is then applied to the slide and let to sit for about 1 minute. The final step is rinsing off the gram safranin. Upon rinsing, the E. coli, which is a gram-negative bacteria, will take up the counterstain and appear pink or red underneath the microscope. Staph aureus, being a gram-positive organism, will remain purple upon microscopic inspection. Your next step would be to prepare the microscope using the oil immersion lens. Let's take a look at our results. Our end results show that gram-positive bacteria retain the primary stain and stay purple, while gram-negative bacteria take up the counterstain, gram safranin, and appear pink or red. As we wrap up, let's review the most important takeaways from the gram staining procedure. First, remember that gram staining is a fundamental method used to differentiate between gram-positive and gram-negative bacteria. This helps guide diagnosis and treatment decisions. Second, each reagent in the process plays a unique role. Without all of them, the results wouldn't be accurate or meaningful. And finally, getting the sequence and timing right is absolutely critical. Even small mistakes can change the outcome, so be careful to pay attention to detail at every step. Thanks so much for watching. I hope this helps you feel ready to try gram staining on your own. Good luck in the lab.